Hi, I'm Ava Brandley. I'm going to be presenting on eutrophication and its effect on invasive species success rates. This is the paper I looked at, titled Increased Invasion of Submerged Macrophytes Makes Native Species More Susceptible to Eutrophication in Freshwater Ecosystems by Peng et al. Now we're going to do some background. There's some important terms. Submerged macrophytes are aquatic plants with vegetative parts that are predominantly submerged under the water, and the plant is rooted at the bottom sediment or soil of that body of water. We also have three different submerged macrophyte species. H for ticulata and P Giannis are the native aquatic plants that were looked at in the study, and E. nataliae was the invasive aquatic plant species that was studied. And that's actually a plant that's native to North America, but because the study was done in China, it's actually the invasive species for this study. So like I just mentioned, the study took place in China. The researchers were really curious because previously, the studies that have looked at the effect of excess nutrients that then lead to better conditions for alien species, which ultimately lead to less biodiversity in the aquatic ecosystems, have been predominantly done looking at vertebrates and invertebrates, and not much has been done to examine this same pattern, but in aquatic plants. And the researchers wanted to look at how the combination of invasive species and other factors, specifically eutrophication, affect the success rate of the native species. And then here we have just a picture of the species that are going to be referred to throughout the study. So we have our native species, and then our one invasive. So the hypothesis was a two-part hypothesis. The first part being that high invasive density with a high eutrophication level can have a significantly negative influence on native species due to strong interspecific competition. And then two, synergetic effects of high invasive density and high eutrophication levels lead to changes in dominance patterns due to species-specific responses. Then they had this nice little graphical abstract. So it just kind of does an overview of their setup and their findings. Those major findings being that high nutrient concentration inhibit the growth or did inhibit the growth of all three submerged macrophyte species. However, due to the broader niche that the invasive species occupies, it was able to do better than either of the two native species in those higher nutrient concentrations. Also, the increased density of the invasive species, E. nataliae, delayed the growth of the native species, H. verticulata. In the high invasion under high nutrient concentration combo led or can lead to a shift in submerged macrophyte species dominant patterns. So pretty much as the nutrient concentration increases, the better the invasive species will do, and the worse the native ones will do, and the invasive will be able to take over dominance in that area. So the materials and the methods that they did was taking samples of all three species from Shahu Lake in China. The samples were all cleaned and cut into 10-centimeter segments, then planted in basins with 7 centimeters of fine sand at the bottom. To simulate the invasion process, they did 3 different densities of E. nataliae, and like contrast to the other two. So they had a low invasion level, which was a 4:4:4 ratio. And they had a medium invasion, which was 6 parts invasive and then 3 parts of each of the native species. And then they had a high invasion setup where it was 8 parts invasive and then 2 parts of each of the native species. And the different plant ratio containers were then divided into different nutrient ratios. So, or like nutrient conditions. So there's low nutrient, medium nutrient, and high nutrient conditions. And then the nutrients that they were focusing on and manipulating in this study were nitrogen and phosphorus. And then after a month of the aquatic macrophytes being in those conditions, they then dried and weighed all the samples. They also quantified the length, the chlorophyll A amount in the plants, the soluble sugar, and then the relative growth rate of all of the samples. And then this data was cleaned and processed, and the main statistical tests that they ran were ANOVA tests. And we have a nice little figure from them just showing kind of how they set up their experiments. You can see that they had like a scale from low to high nutrients and then low to high invasion rate or density. And there was a gradient within that. The main results that they found were that the nutrient concentration significantly affected all the measured traits of the 3 tested macrophytes, including dry weight, total length, chlorophyll A, and soluble sugar, and total community biomass. All that with the exception of the chlorophyll A concentration of P. maculis. That wasn't actually affected by the nutrient concentration negatively. On the other hand, the invasion density only significantly affected chlorophyll A of P. maculis and the soluble sugar of H. verticulata. So the nutrient and invasion concentration interactions only significantly affected the dry weight of H. verticulata, the chlorophyll A of E. nataliae, and the total community biomass. And although all 3 species were negatively affected by the nutrient concentration, the negative effects were apparent for P. maculis and H. verticulata at the medium condition, while E. nataliae wasn't really affected until it hit the high concentration of nutrients. So it had a higher threshold, the invasive species did. They also found that the nutrients could promote chlorophyll A in all 3 species. And then here was their table that just kind of summarized everything, like all the different factors they looked at. And then as far as the discussion, the researchers emphasized the synergistic effects of the invasion and eutrophication levels or concentrations on native submerged macrophytes. The results showed that high invasion density of the invasive species in combination with high nutrient levels significantly inhibited the growth of both native species, but it was particularly apparent in H. verticulata. The study showed that invasion density alone without the eutrophication levels didn't have a significant effect on the biomass and the relative growth rate of native plants, or the native plants. The study also highlighted that invasive species with a wide ecological niche, like E. nataliae, can outcompete native species under high nutrient concentrations. And those are all like important findings, and overall this paper was just important because due to increased human activity like global trade, agriculture, planting, etc., invasive species have been found widespread throughout the world in a variety of different habitat types, and factors like increased CO2, nitrogen deposition, and change in temperature due to climate change all provide opportunities for these invasive species to really establish and be able to eventually outcompete the native species of the area. And the authors really spotlight the importance of understanding the interaction between invasion density and nutrient concentration to protect and restore freshwater systems effectively, because as we have repeatedly learned throughout this course, the best way to tackle freshwater ecosystem protection and restoration is with a holistic approach. So you want to look at more than one factor. Can't just control one thing and expect like the whole ecosystem to figure itself back out. And the authors just really wanted to emphasize that throughout the paper. And then this was just the final citation.